so Yugang, do you want to turn on, do you want to ask your question personally? You can turn on your mic. Okay, I'll ask Yugang's question on his behalf. So he says, hi, my question is regarding the use of wax for organoids. Can you simplify and explain it a bit? So anyone from Dr. Indumati's lab, if you'd like to take that question.
Okay. So hi, so the use of wax is done so that the organoids can be preserved for a longer time. So if we use just the optical, just the
So your voice isn't very clear. Could you? It's breaking up a little.
is it better now? So we use wax for longer storage of the organoids. So not even organoids, for other tissues also, this is a common practice in pathology, like if some biopsy sample goes and or some other tissue sample goes. So this is the practice which is used because this will stay, this can store the tissue for a very long period of time, and therefore we can check whatever we want at even after 2 or 3 years or even 5 years, the sample will be preserved.
Okay, so the next question comes from so India Bioscience has also joined. I'm not sure who's behind the camera, but if you'd like to come and ask your question, or if you'd like me to ask your question, let me know.
Yeah, Anushka, it's me, Karishma.
Oh, hi, Karishma. Thank you for joining.
Welcome. I'm sorry, I didn't realize that I was through the IBS mail. But yes, that's my question, and wonderful episode. I enjoyed it both from the India Bioscience point of view and from my own science point of view. So it's a double joy. And of course, another angle is that Abhishek and Nizam worked with us at Savitribai Phule Pune University. So triple joy.
Yeah, so like if you guys remember, we did episode 1 with Dr. Karishma, so she's joined us here. So Karishma, if you could, why don't you go ahead and ask your question?
Yes, I've seen a lot of interesting organoids being showcased here today. I wondered if the scientists working on them could tell us what's the lifetime. Like, can they actually be stored even for 2-3 days? Can we do sort of time experiments with them? What's your experience been?
So yes, they can be stored for a long period of time. Actually, initially when we get organoids, it's around 3 to 4 weeks of time we need for the organoids to emerge. After that, we need to culture them for 3 to 4 months more so that we get the mature cell types. And we can actually extend them in culture for 6 months also. And we can perform experiments, whatever kind of experiments we want, we can perform with them.
Oh, okay, that's wonderful. Very good to know. That's great. So long-term infection experiments are possible. Wonderful.
Yes, they are possible.
Thank you.
Okay, thank you, Karishma.
Okay, that's wonderful. Very good to know.
Yeah,
that's
thank you
great.
so much.
So long-term infection experiments are possible. Wonderful.
Yes, they are possible.
Thank you.
Okay, thanks for your question, Karishma. Okay, thank you, Karishma.
Okay, that's wonderful.
Oh, I'm so sorry. I could hear the YouTube live. That's why I got confused. Okay, I'm very sorry about that. We'll move on to the next question. from Anshu. If you want to unmute yourself, you can go and ask your question.
Hello, am I audible? Yeah, I would like to ask that which kind of organoids we have prepared till now in the lab? Just like which organs?
Do you? Sorry, yeah, go ahead.
Hi. So actually in the lab, we are basically working on ocular models. So in the lab, we have till now generated retinal organoids and corneal organoids by differentiating these induced pluripotent stem cells.
And what are the difficulties that you usually face while handling organoids? Sorry.
So actually the iPSCs are little bit very sensitive cells. So when we maintain them, if you like triturate them hard, they have the tendency to differentiate. So you have to be very careful while handling them. And when you maintain, because these are the long time cultures, so you have to handle them very carefully. And the supplements which you are adding, which are promoting the generation of these organoids, so the concentration also matters. So they will be like in the adherence, we are normally maintaining these cultures for almost 30 days. And after that, we are scooping them and putting in the suspension to get these organoid structures. And in the suspension, they are maintained further for different experiments. So yes, and while changing the media also, when they are in suspension, you have to be careful because the tips which normally 1 ml tips are very narrow. So normally what we do is either we cut the tips and make it a little bit broader. So if the organoids come inside also, they will be fine. They won't get harmed and doesn't get triturated. So basically these are the things which we take care of during culturing them.
Okay, thank you for your question, Anshu. The next question is from Yogita. Yogita, if you'd like to go ahead and ask your question. Okay, she's asking it to be read out. So I think she has two questions. First is, what are iPSC colonies? And the next question is, what else can be studied using retinal organoids other than retinal blastoma, and what are the possible outcomes of the study?
So the iPSCs are like embryonic stem cells. You guys have heard about, like they have immense power of differentiating to any cell type. So unlike embryonic stem cells, these iPSCs has generated from the adult cells, like from the skin, as you all know. So after that reprogramming, that will bring it back to its pluripotent stage, where they have this property for the differentiating into the multiple lineage. So since our lab is working on the ocular model, so we have differentiated them into the ocular cell types like retinal organoids and corneal organoids. And apart from retinoblastoma, there are several genetic disorders. So patients, we have different patient lines and we differentiated them into the retinal organoids. So we compare what could be the differences between the different genetic status between the control and the patient by using these retinal organoids as a model system in the lab. So like retinitis pigmentosa, Stargardt's disease, I myself work on Leber congenital amaurosis. So these are the different congenital diseases which can be studied using these retinal organoids.
Okay, thank you for your question, Yogita. Next, we have a question from Kasturi. Kasturi, if you could unmute and go ahead and ask. Or if you want, I can read out your question. Okay, so this is for Abhishek. Kasturi asks, can you tell us about why you enjoy working with spheroids?
Yeah, so the first of all, the spheroids are like the easiest three-dimensional model to work with. and they can be fabricated with the cell lines that are routinely used routinely used in any of the lab. So you don't need to go to getting don't need to go and get any patient's sample. For that, you don't need any ethical permission to be there. So it's really very easy to work with, and without any ethical barriers, or without any separate permission required. Moreover, these can be formed with a single type of cells to make them very simple, and to make data very understandable. And then you can also form them with by mixing various cells together and make a complex types of spheroids, so that it will mimic in vivo tumor like structures as well. And nowadays lots of papers are coming up with the heterospheroids, where people are mixing different type of cells. They are making it very complex, and they're using it as a drug screening platform, as well as they're using it to understand uh, different basic biological questions as well. So this model is useful in a applied way, as well as in basic biology vision as well. Moreover, you can regulate their size. They are very much amenable for laboratory conditions and laboratory manipulations. So you can design spheroids of different shapes and size depending upon your purpose. So, and along with that, they are very much resource friendly. Like you don't need to use various growth factors or costly medias in order to work with spheroids. So even in a very basic lab setup, you can also address very complex questions using a very simple model like spheroids. So that's why I enjoy working with spheroids a lot. I hope I answer your question.
Okay. Yeah, thanks for answering that, Abhishek. The next question, so Yugang has a comment, or just I think a follow-up question. So the tissue would remain adherent to the wax and won't affect the morphology. So Yugang, I think you have a follow-up question. If you want to go ahead and ask that.
Yeah, I have a follow-up question. So regarding the use of wax, basically, so I basically had the question that would the morphology be affected at all? Because once we use the wax for basically culturing of the tissues, and then we are basically slicing it and plating it. So at any point of time, is there any concern regarding the structural integrity or morphological factors? And the second question was, how are Yamanaka factors induced into the cell lines? Like what are the techniques and the methodologies used for that?
So yes, wax does not affect the morphology of the tissues because we are using hot wax. It is at around 65 degrees Celsius. So the tissues will the wax will penetrate in the tissues as well, and therefore they do not fall off, or their morphology is not affected with wax. And for your second question, yeah, Yamanaka factors are nucleoaffected. So there are various ways. You can either transduce the Yamanaka factors, you can make the viral soup and then you can transduce, or you can nucleoaffect them using a nucleoaffector machine. So that is the integration-free method where you use the episomal Yamanaka factors. Or more recently, one more technique has come, that is usage of the constructs which are used for the Sendai virus kit. The Sendai virus kit has come where the virus is used, but it won't integrate into the host. So there are various methods. These all methods you can use for generation of iPSCs.
Oh, okay. Thank you.
Okay. Thanks for your question, Yugang. Next, Anshu, I'll get to your question in a bit. Maybe we'll go to someone who's not asked a question yet. So Khushi, why don't you go ahead and ask your question? Or if you want me to read it out. Okay. So Khushi has asked, can mesenchymal stem cells be used instead of iPSCs?
Hi. So the mesenchymal stem cells can't be used instead of iPSC for generating retinal organoids, because mesenchymal stem cells can differentiate into the limited lineages like chondroblast, osteoblast like a bone cells, and adipocytes like fat cells. But iPSCs can differentiate into any cell type, like you can make hepatocytes, cardiocytes, even these chondrocytes, osteoblasts, everything. But since we are focusing on, as I said, ocular diseases and ocular disease modeling, so that's why we are differentiating these iPSCs into the retinal organoids and corneal organoids.
Okay, I hope that answered your question, Khushi. Next question. Okay, we'll go back to Anshu. So I'll just read it out because it's a short question. What are some of the difficulties in handling these cells in the lab? So organoids and spheroids. I think both of you guys can, both labs can answer what are some of the difficulties they face in handling these cells. Sudipta, you're on mute.
So I think I have answered this question about the difficulties, but still I can go once again. So for the difficulties per se, as yes, it's a little time-taking process. You have to be careful while handling these cells during from the reprogramming till you have a stable iPSC colony. It's a tedious process, but once you get, yeah, they are a little stable. But while handling them, you have to handle them carefully, not triturating very hard. Otherwise, they tend to differentiate. And you actually want the undifferentiated population to differentiate into particular cell lineage. So that is one thing. And during the maintenance, as I already told, this is a long-term cultures. While handling, you have to be like particular about on which day, which kind of a growth factor you are adding, and what concentration you are adding, what is the proportion of media changes, and all those things matters for this organoids, getting these organoids into the culture. And once you get it, scooping and putting in the maintenance in the suspension, because since there is no other, so in case of the retina, the photoreceptor are supported by RPE layers, but here we don't have like RPE layer attached with them, which provide them nourishment. So from the media itself, they get it. So you have to be carefully changing media using these, because the 1 ml tip, as I said, it's a narrow. You have to cut them, make a little blunt, and then use it for media changing, because once they get inside of this tip, narrow tip, and you will push it out, they will get, the morphology will go off. So basically you will lose that morphology, and once they will lose the morphology, these are the connections, interconnection between the different neurons. And once it goes off, it takes a lot of time, or otherwise they will shred off. Basically, they will die once the connection between the neurons goes. So that's the thing. So you have to be careful while handling this. So this is from my part. Maybe Abhishek can add from his side.
During working with spheroids, these are like around 400 to 500 micron small structures. So it's very extremely difficult for them to visualize using a naked eye. But whenever you are doing or handling spheroids, you have to be very careful in a way that they may, most of the times they stick to the pipette tip, as Sudipta has already mentioned it. So either you need to use a wide bore tip, or we generally you cut the tip and then harvest the spheroids. Along with that, because this is and you can generate spheroids based with using different cell lines. So to fabricate spheroids using different cell lines, you have to standardize the protocol. Few cell lines are very amenable to form spheroids, and other cell lines, they are very difficult to form spheroids. So sometimes you need to add some external factors to like some extracellular matrix protein in order to get them together and form a clump-like structure. And few spheroids can be formed within like 3 days, and others may take time up to 7 to 8 days as well. Along with that, these being a very thick sort of structures, visualizing the interior of these structures, it's quite difficult. Like, for example, we have visualized the actin, cytoskeleton, and nuclei from these spheroids. So these can be visualized using simple confocal microscope, and we can go up to the depth of 100 to 200 micron. But if you want to see the interior of these spheroids, you may need some high-end microscope like multiphoton microscope, or even you can go for IHC, like embedding them in a wax, as I've already shown in the video as well. But because of their very small size, it is really difficult to embed them into wax and then section them. So we are still dealing with all these things. That's it.
Okay, thank you guys. We'll go to the next question. So Pratiksha has asked, or Pratiksha, if you want to, I think, okay, I'll ask the question. What split ratio do you use for iPSCs?
This split ratio, the split ratio that we use for culturing iPSC is 1:6. So if we have one well of 6 well which is confluent, so this can be split into the other 6 wells. I mean, one well can be split into 6 wells of a 6 well plate.
Okay, next question. So is another question from Yugang.
Yeah, hi. So my question was regarding spheroids, basically, that if we are making in vitro spheroids, so in that case, if at all the uncontrolled growth has to be managed for a particular cell type, as the culture becomes more complicated, how can we approach regarding that particular aspect? Because in the end, uncontrolled growth can become problematic.
Because these are a tumor model and tumors are inherently dividing uncontrollably, it is not right or it is not correct to manage their growth. We'll have to let them grow as they want to, because we want to mimic the in vivo scenario. So we are not going to manage their growth, but in case of they are overgrowing or exhausting the medium, then you just need to take care that they are having sufficient number of like nutrients are being provided in the cultures. So either you need to give a media change to them, or you need to replenish the medium, and any growth factor is added, if any added. So that's it. So we don't need to manage their growth, but we are going to maintain the nutrient balance.
Okay, so we'll continuously keep culturing them. We'll not disrupt their uncontrolled growth.
Yes.
My question was
yes.
basically regarding that because while we are culturing them in vitro, so they will be capacity constrained. Like in a particular well, only up to certain level they can uncontrollably grow. Then they will exhaust all the resources. So either you transplant them or you perhaps increase the media. That was basically the issue. So we'll continuously keep on culturing them.
Yes, yes. So you give a media change, and it never happens that they outgrow a lot, because it's a 3D structure, and it's very difficult for the nutrients to penetrate inside the spheroid. So interior of the spheroid, there will be generation of a necrotic zone. So many of the cells that are present inside the spheroid, they will die. But that is also in a way very important, because in vivo tumors, they also have a necrotic zone. So that is also getting recapitulated using this spheroid model.
Okay, thank you.
Okay. Next question is from Nanika. If Nanika, if you're here, you can go ahead. Nanika, are you here, or would you like me to ask your question? Okay, I'll just go ahead and read out the question. So Nanika has asked, what growth factors have you used for iPSC production?
So there are for iPSC production, there is no specific growth factors as such, but we use iPSC essential 8 medium for culturing iPSCs, which is supplemented with BFGF, that is basic fibroblast growth factor, and sometimes we supplement it like extra supplementation of this BFGF is also being given in the growth medium.
Okay. Next question is from Kadambari. So Kadambari, do you want to go ahead and ask your question, or do you want me to read it out?
Hi. Sorry for that. So can you explain the difference between culturing spheroids in an adherent and suspension culture, like the difference?
Okay, so spheroids are generally cultured, always they are cultured in a suspension culture. So we don't want our cells to go and adhere to the plastic surface. So we are preventing their attachment using an agarose coating, or there are commercially plates available which are called as ultra low adherent plates, which will not let the cells attach to the surface. And I have never seen anyone growing spheroids in adherent type of culture. So in case of cell migration assays, we are transferring these spheroids which we were growing in suspension. So we transfer them into an adherent plate, so that there the spheroid will, now the cells in the spheroids will be able to attach to the surface and then migrate. So it is simply very similar like an scoop of ice cream that is melting. So it looks something like that. So spheroids are always grown in suspension, and whenever we want to do cell migration assay, then that time we will transfer them into adherent type of plates.
All right, okay. Thank you.
Okay. Another question. So we have three more questions. So we're running a little bit late, so I'll just go ahead and ask the questions. So Young has asked, apart from drug testing and screening, are cell therapies also studied in these models? So both of you guys can go ahead and answer this.
Okay, till now, spheroids have been widely used for drug testing and screening. But yes, nowadays, people are trying to use these spheroids for screening cell therapies as well. So, CAR T cells are have a very wide success in treatment of leukemias or blood cancers. So similarly, even our lab is trying to use this model to screen cell therapies similar to what CAR T cells do. We are trying to develop CAR macrophages as well in our lab, and we are going to screen their activity using this very simple spheroid model. And there are different types of inhibitors that also can be studied, as well as these can be used to study interaction between different types of cells. Right now, we are studying interaction of tumor-associated macrophages with the cancer cells using spheroids. You can also study interaction of the other different types of cells like fibroblasts or even mesenchymal stem cells or adipocytes or any other type of immune cells with their interaction with the cancer cells. So these all questions can be answered using spheroids as well.
So in case of iPSC derived retinal organoids or RP, basically they are in the clinical trials right now. So yes, there are cell therapies are going around the world where they are basically focusing on the outer retinal part. So the retina is basically made of different layers. The outermost layer is the retinal pigmented epithelium. Next to that, that is the photoreceptor. So in case of any genetic diseases, these layers get affected first. So in the clinical trials right now, so people around the world, the scientists are culturing the retinal pigmented epithelium as a sheet or like a cell suspension. They are directly trying to inject into the subretinal space in the eye, and where they are proposing that these will take over the cell function, like form a sheet-like structure and get associated with different layers, and will serve that function and can restore the vision till some extent. So all these studies are going on in per se for the iPSC derived cell therapy.
Anushka, you are on mute.
Oh, I'm so sorry. I'll ask the question again. So when you differentiate iPSC cells into ocular cells, how is this process governed, and what differentiation factors would be required to change them into these cells?
So the differentiation means like you are supplementing the cells with different types of growth factors at different time points. So for these, basically, they pushes this iPSC to a particular lineage. So as I said, since we are working on the eye, so we are pushing them toward the retinal organoids. I said, retinal lineage. But yes, there are studies where people use different growth factors and they differentiate them into the pancreas, liver, and other so there they are supplementing with different growth factors, and that is basically pushing them toward the particular lineage. That's what I can say. And it is also covered, we have recently published in the manuscript, and there are different papers available. They talk about different growth factors can be used for different cell type to get in the culture.
Okay, and last question from Yogita. So under wax infiltrated microtomy technique, the cells under study are not living since they are fixed. How do you culture them and observe the cell differentiation and lineage under the microscope?
So I actually, when these organoid is there inside the wax, so we don't study the same organoid for different time points. So suppose we are checking the organoid at day 40, day 55. So till day 40, the organoid was in culture. At day 40, it was fixed. Then it was processed under wax. So wax won't affect, it will not affect anything of the cell. You can study all the proteins, their localization, their interaction at that time point also. So have I answered?
Different time points, early and late.
So actually you can fix the organoids at different time points and you can study the organoids at that particular time point.
Okay, one final question for Abhishek. Do all cancer cell lines show metastatic behavior? This question is from Nainika.
Hi, Nanika. That's a very good question. And to answer your question in a single word, it's no. Because few cell lines, they have been isolated from a primary tumor. So primary tumor is something like which is present at a one side. So as we are working with breast cancer, so these primary tumors, you will find it in just breast. So they have not spread it to any other organ. So if you have established any cell line from a primary tumor, it will not show metastatic behavior in vivo or in vitro as well. But if you have isolated any cell line from a metastatic site, that means in a body, if the cells are already having inherent nature of metastasis, then only these cell lines will show metastatic behavior in vitro and in vivo. So in our cases, we have used two different cell lines. So MCF7 and 231, both are invasive breast carcinoma, but MCF7 is a non-metastatic cell and 231 is a metastatic cancer cell. So only metastatic cells will show metastatic behavior and non-metastatic cells will show non-metastatic behavior.
Okay, so I think that's all the questions, and we're also out of time. So thank you guys for sitting with us, for your patience. Um, we hope that everyone who joined in to watch the video enjoyed it. And if you have any more questions, you can reach out to all the lab members individually. We'll be able to send you their contact details if you have any follow-up questions. And just to address some questions about the assignment and the B2F circle. So we'll be announcing the date for the B2F circle soon. So in that next episode, you'll get a chance to interact with Dr. Indumati and Dr. Tuli and show them your assignment answers, discuss the answers with them, and have a little bit of a more detailed chat about the video. And we have removed the deadline for the assignment. So the deadline will be open until the end of the Back to the Future series, so that everyone can participate for a chance to come visit the lab. If there are any other issues with the links, I think all the links should be working now, and the assignment link is also open for submissions. So you can go ahead and start working on that. If there are any issues, please send us an email. And thank you again for joining. Thank you everyone from both labs for taking time out this Saturday morning. And we'll see you in the B2F Circle episode next time. Okay. Bye.
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 Thanks.
Bye bye.
Bye everyone. Thank you.